So we've mentioned cytokines as an example of some chemicals that are released. What you have to recognize is that the surface receptors are going to be specific to the chemicals too. And so certain chemicals will only bind to certain surface receptors, so they'll only bind to certain cells. So cytokines can activate some cells and then maybe not activate other cells that are lacking those surface receptors. Also recognize that it is those surface receptors that bacteria and viruses are taking advantage of. Those surface receptors are there for other purposes, but the bacteria and the viruses have molecules themselves that are able to recognize those surface receptors and bind to them. We mentioned adhesion molecules. Those are the molecules that endothelial cells have that they serve to grab onto monocytes and neutrophils, slow them down so that they can move into the tissues. I've mentioned toll-like receptors a couple times already. Many cells have receptors that are able to recognize bacterial products and send out messages. The complement system. This is a system of proteins that can do a number of things. But one of the things that it does is it enhances our antibody response, our immune response. So complement can bind to cells invading bacteria and things like that and serve to flag them for macrophages, for neutrophils. So once they come in contact with something that's been bound by complement, they'll engulf it, bring it in. Complement can serve in the chemotaxis part of the process in attracting macrophages to an area. Complement can serve to punch holes into the membranes of bacteria, gram-negative bacteria, punch holes into that outer membrane there. You should understand the process of phagocytosis. I won't go into it here, but there's a series of steps that takes place: attachment, engulfment. Recognize that there are really only two white blood cells that are involved in phagocytosis, okay, as part of destruction of the cells. Clearly, the B cell has to take in the antigen before it can present it. The dendritic cells have to take in the antigen before they can present it. So there is some engulfment in that sense going on, but it's not phagocytosis. It's not engulfment for destruction. It's sampling these antigens to present to T cells. Inflammation. Be sure that you know the process of inflammation, the steps of inflammation, and the signs of inflammation, or what's causing each of those signs. So redness. What's causing the redness of inflammation? Blood flow to the area. Blood flow to the area because when you were damaged and the bacteria got in there, a release of cytokines caused a dilation of the vessels in that area, and so you got increased blood flow to the area. That causes the redness that we see, and with that increased blood flow, we also get heat. More blood flow, more heat, and some of the chemicals that are being released at the time are also generating some heat. So you've got the increased blood flow and chemical release responsible for the heat that you're seeing there as well. Then you start to see swelling occur. The swelling comes because the fluids from the circulatory system start to move into the tissues. We get neutrophils to move into the tissues, we get macrophages moving into the tissues along with fluids, and so you get swelling that occurs here. This is going to serve a couple of purposes. it does allow for delivery of those cells and proteins and stuff necessary for combating the microbes. But in addition to this, now you're going to have all this fluid in the tissues, and the fluid in the tissues is going to have to be removed, and it's going to flow into the lymphatic system, which means that the bacterial products and stuff also will flow into the lymphatic system along with the dendritic cells that are there. So they'll flow into the lymphatic system as well. So the increased fluid in the area, the removing of it as it moves into the lymphatic system will serve to deliver antigens to B cells that are in the lymphatic system. And so those B cells then get exposed to the antigens, for which they can then go and expose or present to T cells. So it aids in getting the antigens to the B cells. So you've got swelling that goes on there. The swelling then also is going to start to put pressure on the nerve cells that are there. And so you're going to get pain develop from that pressure. There are also being chemicals released, pyrogens, that can serve to enhance that pain, if you will, or increase that pain. Yes?
When the tissues, when the epithelial cells start to spread, what's that process called?
Diapedesis is the movement.
That's the movement of the monocytes through, but what's the process of when the epithelial cells actually start? It's the cytokines that cause that separation?
Mm-hmm.
When they start moving. Is there a name for that?
What's that?
Well, I was studying that today, and the mast cells release histamines that then make the epithelial cells spread, and then all the fluid leaks in, and she's talking about.
She wants to know the term for the movement of the endothelial cells apart.
I'm actually wondering what she's saying, mast cells, that you're saying inflammation. Now, aren't mast cells like a histamine?
Yeah, mast cells and basophils, they both release similar molecules. Basophils and mast cells, very similar.
Okay, so that's not just an allergic response, that's actually an inflammatory, like a puncture wound or something. You'd also get histamines.
Yes, you get histamines released in a hypersensitivity response,
right.
You know, in an allergy. You'll also have histamines released in an acute inflammation response.
Good, thank you.
Yes. What's the benefit of a fever?
Bacteria.